First of all, I would like to start with this, what is known about nuclear mechanics and dynamics. There is more and more evidence that mechanical clues, either in animal or plants, are very important to regulate development and gene expression. But in animals, there is a clear evidence that nuclear and dynamics and mechanics are important to drive this regulation. But these underlying mechanisms are still poorly understood in plants. So it seems that nuclear shape change is a biological phenomenon that occurs during developmental and in response to environmental stresses. Yep. So what we are interested in, what is the response of the mechanical stress reaching the nucleus? And we know that the nucleus is surrounded by an environment called the nuclear envelope, which is related with the cytoskeleton, actin, and microtubules. But inside the nucleus, close to the inner nuclear membrane, we have also a nucleoskeleton in plants, with specific proteins which form some laminar organization with some well-known reticulation of the network. But how is the response at the nucleo-cytoplasmic continuum, and how it will impact on cell fate and plant stress response? We know also that there is some outside-in signaling and inside-out signaling. But what are the mechanisms behind? And now we would like to answer this question from tissue to single cell levels. So what we know also that nuclear mechanics are very important to regulate gene expression, so that we know it's really well known in animals that under stress you can induce chromatin condensation, chromocenter clustering, or changes in epigenetic marks from with changes in localization of repressive histone marks, but also release of transcription factors. Now going to the nuclear envelope in plants, we know that there is some well-defined link complexins that have been well characterized in clear connection with actin cytoskeleton and chromatin thanks to nucleoskeleton, that is some specific proteins grounded 1, 4, and CACU4. But also we have identified in our team small gene proteins which are connected on the outer nuclear membrane with microtubules, but also connected with chromatid at the centromeric level. And interestingly, mutation of some of these partners at the nuclear envelope have tremendous effect on developmental and nuclear shape and gene expression. For example, the recently discovered PNT2 proteins, which are small mutagenic families, show that where mutated when the mutation of all partners, PNT2, this induces a defect in nuclear shaping in higher order chromatin organization, but also constitutive activation of abiotic stress responsing. So showing that the nuclear envelope is a very important site for nuclear stress response. And also it's induced developmental defects. Previously, we have also shown in our team that the knockdown of the GIP1, G2 genes induce disorganization of a microtubule network, a disturbance of recruitment of microtubule nucleation complexes, but also defect at the centromeric level in terms of loading of synastry at the centromeres, but also cohesion defects. And in addition, we really observe also a defect in nuclear shaping, in increased nuclear rigidity in terms of mechanical properties of the nucleus, with a constitutive induction of touch responsive genes and also induction of developmental defects. So I show you now some example we have investigated upon the mechanical stress in a tissue context. So we have focused our study on the root meristem, and we have used experimental stress using hyperosmotic stress response. So we show nicely, this is a labeling of the nucleus with the inner nuclear membrane, SUN1 GFP. So in the meristem, the nucleus are very rounded, but under hyperosmotic stress, you observe a nice change of the nucleus. And this is nicely correlated with decrease in nuclear circularity, nuclear area, as well as nuclear sphericity and nuclear volume. So so there is a huge change of this nuclear shape under mechanical stress mimicked by hyperosmotic stress. We have also demonstrated that the change in nuclear shape has some threshold depending on the concentration of the mannitol that we use for this hyperosmotic stress. Interestingly, as well, this change in nuclear shape is reversible upon stress recovery. That is illustrated here. Here we have a deformation after mannitol treatment, and after recovery, after 7 hours, you can recover the shape of the nucleus in terms of nuclear circularity and area, but also in terms of mechanical properties. So you can see we have increased rigidity with mannitol and its rescue after stress recovery. So it seems that nucleus really act as a rheostat in plant development. So it affects growth, shape of the nucleus, but also gene expression. And we have demonstrated also that the G proteins located at the nuclear envelope periphery could be important to regulate this process. So definitively, in a tissue context, we have the first arguments to show there is a nice correlation between nuclear shape and nuclear rigidity linked to gene expression and plant development. But we are really interested now to go further in our study and to really understand what is the relationship between cellular and nuclear morphodynamics, what are the impact on development at the single cell level. And for that, we have investigated the root R, which is a perfect model to study nuclear dynamics. Why? Because it's a tubular extension of epidermal cells in specialized cells called trichoblasts located in the differentiated area of the root. There is a polar tuberose linked to this development of the root air. And also root air is very important because it's important for interaction with the surrounding environments in the soil.
Most widely used city gametes rank at like 1.
Sorry? Okay. So to address the question, we have developed in collaboration with Atefas Nasios and Michael Rickling in Strasbourg, a Coverslip-based microfluidic device, which is based on PDMS material for microfluidic chips. And it's made of polydimethylsiloxane, PDMS, which is a mineral organic polymer with a flexible polymer chain with a high level of viscoelasticity. And of course, these benefits of these products, it's a cost-effective, it's already known, and there is some previous studies that have used these devices to analyze plant growth and so on, and but also it's deformable, and importantly, it's gas permeable, which is very important to allow root growth nicely. So this is the basis of our device. So we have 3 independent canals. When we can put the plants in this hole, and you have a detail of the chamber here. So you have a total chamber volume of 6 microliter. The total length is 1.6 centimeter, and the width of the main canal where the main root can grow, it's a 250 micrometer and depth of 100 micrometer. And we have lateral canals of 400 micrometer length with 20 by 20 micrometer width and depth. So this device is based on multi-layer devices, and this elastomer is covalently bound to a glass cover slip. So initially, what is the experimental procedure? we put seeds on agar, which are filled in small tips, and these tips are allowed, to grow in a petri dish for a few days. And when the root reaches the end of the tip, we transfer this tip in our device, you can see here, and with an angle of 45 degrees at the intri. And nicely, after 4 days in the device, we can follow this. Oh, sorry, it's very sensitive. We can follow the root, the main root growth here, and the development of the root hair on the lateral canals. And here you can see the nucleus thanks to the SUN-GFP, which has we can label the nuclear envelope. So also what is important that we have also added some sugar in the media to have to be really in physiological conditions as classical experiments. And the root growth is under a constant flow rate, which is about 8 microliters by minute. So here you have a cell, you can follow during a 60-minute time lapse the cellular morphodynamics changes in real-time imaging. And this work was mainly performed by my postdoc, Gaurav Singh, in the lab, and also the work of David Pereira as a postdoc in the lab of ATEF in Paris. And you can see nicely the development of the root hair on the lateral canals. And here you have the main root hair. So first, what we were interested in is just to see this, if our device was suitable to perform analysis, and we were interested to know if we can reproduce the data that was already published with more conventional approaches using phytogel to allow root hair growth. So in this movie, we can see there is a 60-minute time lapse, and we can evaluate the speed of the root hair, which is around 1.5 micrometer by minute, which is really in agreement with the published data from Dolan. And you can see that it's quite homogeneous for the position of the team during this time, the time lapse, and here we have the tip average speed. So next, we are interested to understand, to analyze, the nuclear dynamics during tip growth. And we have investigated different parameter analysis. First, the nuclear movements that have already studied by the past, but also in more details that was not so much investigated up to now, the nuclear shape changes in the root hair during the root hair developments in terms of circularity, area, perimeter, and aspect ratio. Here we have an illustration of the analysis of the nuclear movement during root hair. And you can see on this video the nice coordinated movement of the nucleus. So we have investigated the positioning of the tip growth in blue and the nucleus surrounding in red. And we can see, as illustrated on this graph, that there is a constant parallel evolution of the nucleus and tip of the nucleus position and the tip, compared to the tip position. And this make a synchronization between nuclear speed and tip growth in growing root hair. And as illustrated here, we have a tip seed speed of 1.6 micrometer by minute, and the speed of the nucleus is around, for the nucleus 1 in this line, for 2.1 micrometer by minute. But interestingly, when we investigate the analysis of the nuclear movement during root hair growth for a longer time, here it's 150 minutes, we can see that the tip growth is really constant, but the nucleus may go back and forth around its mean position here, with a mean speed of 2.3 micrometer by minute. So we are happy because according to the speed, we were in agreement with the work that had been already published by Ketla by the past. But now we would like to really analyze this nuclear dynamic in terms of changes in nuclear shape during tip growth. This is what we have performed. Here we have the movies. And from this movie, we have integrated these images during 10 minutes and as a Z max. And you can see that the aspect ratio, I will define the aspect ratio as a length divided by the width. And so during the 6 yearly minutes, we have a change of this aspect ratio. After 4 minutes, we have an increase of 38% of this aspect ratio, which correlate nicely with a decrease in area and decrease in circularity here. Just to have a better idea of these changes of the shape, here you have a transverse section views in Z-Y axis, and you can see these changes of the nucleus. So we can conclude that there is a quick fluctuation of nuclear shape in growing root air and a nice correlation between changes in aspect ratio, circularity, and nuclear area. Interestingly, when we look at the perimeter and nuclear volume, there is no so much change. So it seems that there is really a 3D reorganization of this nuclear volume, nuclear shape during root air development. As by the past, it was also described that at least for the positioning of the nucleus compared to the tip was changing according if it was a growing root air or mature root air. And indeed, we can reproduce this data with our device. You can see that the nucleus position to the tip distance is quite conserved in the growing root air, but in the mature root air, it's really position at a random position in the root air. Interestingly, there is no clear differences in terms of area in the nucleus in growing or mature root air. But the most important thing is that the nuclear aspect ratio was considerably increasing by 45% in mature root air. So, as for nuclear positioning in the root air, nuclear aspect ratio is also stage dependent during root air development. And also I would like to pinpoint that during in the mature root air, the nucleus is always going fast in a forward and a backward movement. And the speed of this movement is really higher compared to the growing root air. Here we have an example with a forward speed of 3.2 micrometer by minute and the backward movement with a 4.1 micrometer by minute compared to the growing root air, which is a mean with 2.3 micrometer by minute. So in conclusion from this work, for the first experiments, we can conclude that our microfluidic device allows to monitor in real-time nuclear dynamics during root air development. The nucleus of root air cells is a highly dynamic structure undergoing significant 3D shape changes. And aspect ratio of the nucleus and speed migration change during root air development. But to further explore these microfluidic approaches, we can use, thanks to this device, because I just illustrate this here, when we can change quickly the media inside the device. And you have an illustration here by using a transient flow rate increase during 30 seconds. And here you can see nicely that after 29 seconds, you have a completely filled, the lateral canals are quickly filled simultaneously. So, thanks to this property of our device, we can modulate easily the cell shape and chromatin using drugs affecting cytoskeleton and chromatin, but also we can modulate root hair growth by using bacteria or fungi exudates. As a further exploration also, what we are doing now is just to see the impact of canal geometry on root hair growth and to look for nuclear shape and cytoskeleton reorganization. So as an example here, you have a microtubule in green and nucleus in red. And when the tip reach the restriction in the canal, here it's reduced by twice of the length, so 10 micrometer, we can see a change. I can go to the video, a change in the nucleus, go back, and you have also in the change also in this microtubule network at the back of the nucleus. So the next step in our story now is just to see how are correlated the change of cytoskeleton dynamics to chromatin and nuclear envelope organization. So this work is in progress and it's under modeling thanks to the collaboration we have with Henrik Johnson and Tamsin Spielman in UK. But also we would like to investigate how gene expression is changed and modified during development, especially root hair, but now we are going to the main root, especially during this process of development in the root from meristem, elongation, and differentiated area. And we would like also to know what are the key actors involved in mechano-transduction. So finally, I would like to thank my team, and especially Gaurav, but also Elise, that contribute a lot to help us in our experiments, and Atef Asnasios in Paris, Henrik Johnson in UK, Michael Rinkling in IBMC, and Kentaro Tamura at the University of Shizuoka in Japan. And I would like to thank you for your attention. And of course, new PhD students and postdocs are welcome in the lab. Thank you.
Marie-Edith, thank you so much for the beautiful talk. I really would like to open the floor to question, and I can see that the chat is getting populated. And I don't know, Marie-Edith, if you have a possibility to say, but everybody's communicating with you.
Okay, so I can answer this question. Okay, so why the nucleus is moving? So this is a really interesting story. So we think that this nuclear movement is very important to have a connection, to drive also a connection between the tip and the microtubule network. And this is what we are really want to understand now. And we have some interesting data that will be, I hope, will be published soon. So I think for me, this nuclear movement is very important for tip growth. And there is a nice coordination. I think, of course, it was really described by the past with actin, but now we have evidence that microtubule networks is involved as well.
We also have a question, is asking what would happen if you put the setup, your experimental setup, against the gravity?
So it's a good question, but for the moment we have we are not going in this direction, but this could be performed with our device for sure. Yeah, but I have no answer with this setup.
Marie-Edith, while the chat is being populated, I would like to ask you, how conserved is this change of nuclear shape among plants? Do you have any idea? Is this, don't want to call it specific to Arabidopsis, but how frequent is the nuclear scene to change shape throughout evolution?
Yeah, for the moment, I will say most of the studies has been performed in Arabidopsis. I think there is some work performed in maize, but it was not so much investigated in terms of quantification and the analysis of the shape. But for sure, the shape, this elongated shape is quite common, I think, in plant tissues. at least in differentiated plant tissues, okay? But I think, from my point of view, I think most of the theses, unfortunately, that have been performed, but I hope it will go in other direction. We have some progress in preparation, some programs, in preparation now, just to analyze this process in wheat or in other plant organisms. So using these devices, so that we help to answer your question.
Yes, it does. Thank you so much.
You might also like to look at the intermediate filaments. I'm sorry, but I think that there is no clear intermediate filaments in plants. For my knowledge. And the evidence of the certain with metal all in scientist stress tolerance. Okay, thanks. Metal full sanity tolerance. So yeah, I have some interesting now. Okay, thank you. Just have some information about interaction with metallothionein. Okay, rice filament. Okay. Okay, thank you for the information. For me, it was not clear. These intermediate filaments. Okay. Protective factors and can control some ion concentration. Yeah. So a question from Christophe. Drugs to inhibit some of the putative factors of nuclear migration. So of course we can play with actin, just to play with cytoskeleton drugs, but also with oryzalin to alter microtubule dynamics. And can you also control some ion concentration, calcium at the root tip? Yes, we have some markers to control this calcium flux. So we have some, we can follow this by live cell imaging. Yeah. Yeah. So it's just mutants in filaments in rice. So thank you. Because from my knowledge, I think nothing is known in Arabidopsis. I don't see any papers of that. Okay, just asking some. So it could be good to collaborate with Dr. Parikh because there are already many tools in rice to investigate the role of these intermediate filaments.
Yeah, I think all these are very good questions, and they speak to the fact that your talk was very interesting, Marie-Edith.
Yeah, thank you.
Well, with this, I don't see additional questions, but I think the talk was fantastic. Thank you so much, Marie-Edith, for showing this